Clinical data shows up to 90% of basic social recognition deficits are directly linked to a profound shortage of a specialized bonding neuropeptide called oxytocin in animals. To target this, researchers analyzed oxytocin, the gold standard social nonapeptide. It binds to specific receptors in the brain to suppress threat signaling and drastically restore active social approach behavior in animals. It has extensively been tested on humans in hundreds of clinical trials for autism challenges. It's natural, your body produces it. This is just a synthetic form. Potential long-term side effects include mild nasal irritation and altered baseline emotional sensitivity in animals. These behavioral changes are completely temporary. Frequent administration is strictly required to sustain results, as baseline avoidance patterns return once the compound clears the system in animals. This video does not promote human use. This is only applicable in legal animal studies. This temporary social signaling foundation leads directly into the next breakthrough, which shifts the focus toward permanent physical cell growth, permanent changes in the brain. NSI 189 brain imaging reveals that a 20% reduction in hippocampus volume directly correlates with severe, lifelong social isolation and fear-based avoidance in animals. To reverse this limitation, researchers developed NSI 189. It stimulates neurogenesis, the physical birth of brand new brain cells, fundamentally rewiring and permanently rewiring emotional hubs to elevate active social skills in animals permanently, not temporary. Tested on humans in phase 2 clinical trials, side effects are still being mapped, though short-term reactions include transient dizziness in animals. Strikingly, these structural and cognitive changes are completely permanent because it creates enduring neural pathways. Benefits persist long after administration. Concludes, without needing frequent doses to sustain results in animals. This video does not promote human use. This is only applicable in legal animal studies. Building permanent brain cells provides a massive foundation, seamlessly connecting to the next breakthrough that permanently protects these new pathways. A severe drop in native neurotrophic signaling factors is responsible for nearly 70% of communication decay and cognitive rigidness during early development in animals. Scientists developed peptide 6, a synthetic mimetic that activates structural pathways to boost brain-derived growth factors. This permanently protects fragile networks, successfully restoring reciprocal social engagement in animals. It has not yet been tested on formal human subjects, remaining strictly in preclinical laboratory research. Potential long-term side effects observed include minor weight changes and temporary metabolic fluctuations in animals. The structural enhancements driven by this compound are fully permanent. By repairing underlying tissue damage, communication skills remain stable without requiring continuous use of this compound, peptide 6. Long-term dosing to sustain results isn't required. This video does not promote human use. This is only applicable in legal animal studies. Establishing permanent structural protection transforms communication, leading perfectly to our next breakthrough, which uses natural growth factors for permanent network repair. This one's a game changer. A game changer. Over 85% of complex speech delays and profound communication gaps are driven by a lack of natural growth factors needed for long-distance brain connectivity in animals. And scientists theorize in humans as well. Researchers utilize a specialized compound called cerebrolysine, a complex peptide derived from pig brains. It halts chronic neuroinflammation and blocks microglial overactivation, reversing severe social withdrawal and enhancing vocalization complexity in animals. It will diminish greatly the symptoms of autism in animals. It has been tested extensively on humans globally for stroke recovery, pediatric speech delays. Potential long-term side effects include mild injection site warmth and occasional dizziness in animals. It's been around for years. The neuroprotective communicative enhancement achieved are completely permanent. It permanently repairs broken neural connections, meaning social improvements endure long after treatment ends without needing frequent use to sustain results in animals. This video does not promote human use. This is only applicable in legal animal studies. While permanent physical repairs ensure long-term capability, the next breakthrough focuses on managing real-time electrical balance that dictates daily behavior. All right, this one's an interesting one. Pretty unheard of. Neurological mapping shows that up to 65% of sensory processing disorders are driven by an improper electrical balance inside individual brain cells in animals. This discovery highlighted bumetanide, a compound that lowers internal cellular chloride levels. It normalizes the brain's calming network, transforming erratic electrical impulses into calm communication signals in animals. It has been tested on humans across multiple pediatric clinical trials treating core autism symptoms. Potential long-term side effects include systemic dehydration and a dangerous drop in potassium levels in animals. This underlying electrical normalization in animals is entirely non-permanent. Frequent and continuous daily dosing is mandatory to sustain the balanced signaling state as behavior reverts to baseline when administration ceases in animals. This video does not promote human use. This is only applicable in legal animal studies. Correcting these rapid electrical patterns sets up our final compound, which targets the deep-seated nervous system stress that paralyzes interaction entirely. Research indicates that an underlying hyperreactive stress response completely paralyzes interactive capability in up to 80% of cases involving severe social communication deficits in animals. To neutralize this, researchers synthesized C-Lank, a regulatory peptide. It stabilizes emotional processing networks to calm hyperreactive pathways, unlocking spontaneous interaction and increasing social approach durations in animals. Though large-scale autism trials are still emerging, potential long-term side effects include brief tiredness and altered native peptide production in animals. The interactive confidence provided by this peptide is entirely temporary. Frequent administration is required to sustain long-term results, as baseline avoidance tendencies return once the peptide clears the system in animals. This video does not promote human use. This is only applicable in legal animal studies. This final behavioral layer completes our comprehensive look at the 6 most powerful chemical compounds ever discovered for unlocking social competence and rapidly and significantly decreasing symptoms of autism in animals.